Good morning, everyone, and thank you so much for joining to this third seminar of the Let's Talk Flow series. My name is Andrea, and I work as a technical application scientist in the Flow Panel Design team at Thermo Fisher Scientific. And as you might have guessed already from the title of the presentation today, although the main focus of this seminar series is flow cytometry, today we are going to take a step back from the so-called flow technicalities, and we are going to focus on the biology of our samples and a focus specifically on cytokines. So when we think about a flow cytometry experiment, this is something that is typically not isolated by itself on an island, and rather a flow cytometry experiment belongs to a larger picture. So as you can see in this diagram, typically we have many steps to consider from the point where we start planning our flow experiment until the point when we are ready to acquire our sample, obtain our data, and report our findings. So in the previous Let's Talk Flow seminar, we focused on tips and tricks for our spectral panel designs. And in upcoming talks, we are going to cover other parts of the flow cytometry, larger picture, such as controls and sample prep. But today, we are going to take a step back, as I said before, and focus on the very beginning of the story, which would be the biology of our sample. And specifically, we are going to talk about cytokines. So as you might know already, cytokines are small proteins secreted by cells, and they enable cell-to-cell communication through pathway signaling. And my apologies if I moved the laser pointer too much, but my slides are not moving properly. So my apologies for that. So going back to cytokines, when we think about cytokines in our experiment, we might need to think about them in two different ways. Cytokines might be involved in our experiment as a tool in functional assays themselves. For example, we might want to use a cytokine to differentiate or expand a cell population or to obtain a desired phenotype in our cells. On the other hand, cytokines can be the focus of our experiment as a target for detection. So we might be interested in detecting the levels of a particular cytokine. We are going to divide this presentation in two main parts. And first, we are going to focus on cytokines as a tool in functional assays. So how to use cytokines to obtain a desired phenotype or desired properties in our cell sample, and how to integrate flow cytometry in this type of workflows. To start, we wanted to bring up an example of using cytokines and other recombinant proteins to differentiate our cells. And this is one of the protocols we have standardized at Thermo Fisher Scientific, which is the differentiation of monocytes to macrophages in culture using MCSF. Here at the top of the slide, you can see the role of MCSF in the global macrophage differentiation pathway, starting from progenitor cells and also from monocytes, as we will do in this particular protocol. And in this protocol, what we do is to take monocytes that have been isolated, for example, from PBMC or any other source, and we culture them for 6 days in presence of this MCSF. In addition, we can add cytokines such as IL-4 to obtain an improved viability of our differentiated cell sample. After these 6 days, we will obtain fully differentiated macrophages. To check that we obtained the macrophages we desired, of course, we can check the morphology of our cells directly via imaging, because macrophages will be more complex cells compared to monocytes. And also we can check the phenotype by a flow cytometry. In this example here, you can see the analysis of CD68 expression after culturing monocytes for 4 days with MCSF. So we can perform this phenotypic check after the 6 days, or as we did in this example, more or less at the point of the protocol to check that everything is going correctly before we finished. In this case, we would expect an increase of CD68 expression upon differentiation. Interestingly, We can both check morphology and phenotype at the same time via flow cytometry, and we can do that not only with the scatter signals, but also using a flow cytometer that is equipped with a camera, with imaging features, so we can measure both things simultaneously. I'm not going to enter into details for this seminar, as this is something we are going to cover in the next Let's Talk Flow series. If you're interested in this particular protocol, or if you need any further details about how to perform this differentiation, you can, of course, check the protocol at our Immunology at Work Resource Center, which you can visit at thermofisher.com/immunology. And inside, you will find other protocols that are related to the use of recombinant proteins as cytokines, educational materials, and in general, many information points about immunology in your experiments. The other protocol we wanted to bring out as an example is a little bit different, and in this case, we are not going to focus on cell differentiation, and we are going to focus on obtaining a particular phenotype from our cells. So in this case, we are going to focus on mouse cells instead of human, and we are going to take macrophages again. But we are going to start from differentiated macrophages. And in this case, we are going to use cytokines and other compounds to induce different phenotypes. So roughly, we can classify macrophages into two main phenotypes, which are M1 and M2. M1 macrophages are those which have the pro-inflammatory properties and strong antimicrobial properties, so those that we can classify as the fighting macrophages, whereas the M2 macrophages are those that are involved in tolerance induction. So if we take mouse macrophages, we can polarize them towards the M1 or M2 phenotypes. This is something we can do by culturing the macrophages first with interferon gamma together with LPS for 24 hours. With this, we will obtain the M1 phenotype. Instead of using this, if we want to achieve the M2 phenotype for our experiment, we can polarize our cells with IL-4 also for 24 hours, and we will obtain our M2s. To check that the protocol was performed successfully and we obtained the cells that we wanted, we can, of course, check the phenotype using flow cytometry, in this case, intracellular flow. Here you can see how our M1 macrophages are mostly positive for iNOS, while they are mostly negative with only a small percentage of positive cells for arginase 1. On the other hand, if we check the M2 macrophages, we can see that they are all positive for arginase 1 and none of them expresses iNOS. We will go into details about how to perform intracellular flow in one of the upcoming talks of the series. But for now, we just wanted to illustrate how flow cytometry is essential in this type of protocol to ensure everything works properly with our recombinant proteins. We wanted to bring up a couple of more examples on this regard, and for this, we would like to focus on a field that is very important for our challenges as a society right now, which is immune oncology. As you might know already, immune oncology is the field that tackles the immune system to try to activate the immune response to destroy tumor cells. This is one of the main focuses right now for cancer therapies, and many efforts are focused on stimulating T-cell responses to destroy the tumor cells. In this diagram, you can see the main points of T-cell stimulation and T-cell responses for immune oncology. For example, you might be already familiar with checkpoint inhibitors that can be targeted in the T-cell itself or instead in the antigen-presenting cell. Also, you might be familiar already with cancer vaccines that use tumor antigens to stimulate T-cell responses to destroy the tumor. But today, we wanted to focus on another point of the immuno-oncology field, which is one of the most promising therapies right now for cancer patients, which is the CAR T-cell therapy. So CAR stands for chimeric antigen receptor, and what these T cells have is an antigen receptor that has properties of recognizing full antigens instead of only small epitopes as the TCR does. But this receptor is combined with the transduction signal domain of the TCR itself. So with this, we can take the T cell directly to the antigen we want to destroy, and with this trigger the destruction of the tumor cell. Cytokines are also very important in generating this type of CAR T cell therapy, and today we wanted to bring an example of the role of cytokines in generating stem cell-like memory T cells. So these T cells resemble more naive T cells rather than fully differentiated T cells. As you can see in the slide, they express both CD62L and CD45RA as the naive T cells too. And they have more stem cell-like properties, more self-renewal potential, and more proliferation potential. This makes them desirable when we are developing CAR T cell therapy, and also, interestingly, they are more safe for the patients because they will not be so much activated as a differentiated T cell product, and this prevents exacerbated activation responses that could trigger side effects and excessive cytokine production and activation for the patient. In this case, what we did in terms of Fisher Scientific was to use IL-7 and IL-15 to generate this type of stem cell-like memory T cells, or TSCMs, which will have more stemless properties, improved survival, and less activation. And to check this, we use flow cytometry. And if we go here, we can see that the majority of the cells we obtained with this protocol using IL-7 and IL-15 have these TSCM properties, also with a significant percentage of central memory T cells, with a very low percentage of fully differentiated or effector T cells. When we check the activation profile of these cells, we observe that the late activation marker CD69 was not present in these cells, and there were only a few cells expressing the activation marker CD25. So with flow cytometry, we could check that the phenotype of these cells was the desired one. Another example in therapy, in this case focused on NK cells, is the use of recombinant proteins to differentiate iPSCs into NK cells. NK cells can be also interesting from a cancer therapy perspective because as they are our natural killers, they can also contribute by destroying the tumor cells. So in here, we wanted to highlight how we used multiple recombinant proteins, which you can see at the middle of the slide, to come from iPSCs to hematopoietic stem cells. Once this differentiation was completed, we differentiated the hematopoietic stem cells into NK cells using this set of recombinant proteins you see below. Once we obtained our NK cells, we wanted to check their phenotype by a flow cytometry before we proceeded with the full expansion of our NK cell product. And as you can see below, with this protocol, we obtained a majority of NK cells expressing CD56 with an absence of T cells, which would be here, CD3 positive, CD56 negative, or NKT cells, which would be the double positives. Moreover, with flow cytometry, we could check that these NK cells expressed NK typical markers such as CD16. With this, we knew that the phenotype that we wanted to obtain was successful, and we were ready to expand them using IL-2. Now that we finished with our examples, we would like to summarize everything with a little overview of selection tips for functional assays. And although this is not going to be a comprehensive list, it provides us with an overview that could guide us in our next experiment. So the first thing we need to ask ourselves is, what actually do my cells need? So we need to cover our cells needs to address our biological question. First, we might need to be more interested in antigen-specific stimulation. So, for example, we might be interested in studying T cell responses against a specific antigen, a specific virus, for example. And in this case, peptides could be an interesting option, especially when working, as I mentioned, with T cells. If we are more interested in cell differentiation or cell survival, maybe we might be more interested in using cytokines or growth factors to obtain the desired phenotypes as we displayed in the previous examples. In this case, if we're interested in peptides, cytokines, or growth factors, we can go to the Thermo Fisher Scientific webpage, and at the top of the screen, you will see this little filtering option where you can select proteins and peptides. This will trigger another filtering layer where you can filter through the protein name, the species, the expression system you're interested in, the product type, application, and others. So here you can be ready to select your cytokine or growth factor of preference, and also any peptides you might be interested in for more antigen-specific experiments. Also, we might be interested in a general activation protocol or a stimulation protocol. And of course, we can achieve this also with peptides and cytokines, but there are other products that could help us achieve this type of phenotypes, such as Dynabits or other molecules such as CON-A or LPS, as we saw in one of the previous examples. In this regard, at Thermo Fisher Scientific in the Gibco and PepperTech brands, we have multiple recombinant proteins across 3 different grades. We have options for research use only applications, animal-free, and GMP. And this covers, as you can see here, an extensive amount of applications such as oncology, organelle research, immunology and cell therapy, as we saw before, and also stem cell research. And here you can find multiple types of proteins. You can find cytokines, growth factors, neurotrophic factors, chemokines, or interleukins. So there is a very broad menu to address multiple types of experiments. Now that we covered the use of cytokines as a tool to obtain a desired phenotype, we would like to focus a little bit more in the use of cytokines as the target of our experiment for their detection. This is something we can achieve via multiple techniques. Here you can see an example of how we measured interferon gamma by flow cytometry. Here you can see CD4 T cells in blue and NK cells in purple. As you can see in this histogram, upon stimulation, NK cells were producing interferon gamma, where CD4 T cells were not producing it. When non-stimulated, none of the cell types were producing interferon gamma. So this illustrates how flow cytometry can be helpful for cytokine detection, but there might be other situations that we are going to cover this in the next slides, where flow cytometry might not be the best option. And in this case, we have multiple types of immunoassays to the rescue to help us detect all our desired cytokines. So going back to the previous topic, we can encounter multiple situations in science where flow cytometry might not be the best option to quantify a cytokine or any other protein. And in this case, flow needs a little bit of help from their friends to answer our biological question and achieve our experiment goal. For example, we might encounter situations where the sensitivity of the flow cytometry method is not sufficient to detect our cytokine of interest. Also, it might happen that our target cytokine or protein has been already secreted to the extracellular medium. And in this case, we can't detect it by a flow cytometry as it won't be inside the cell anymore. Although we can block secretion, for example, with inhibitors such as perfalin A or monansin, this might be not compatible with the biological question we want to answer. So we might encounter situations where we cannot use protein secretion inhibitors, and then we cannot detect anything that has been already secreted. Also, we might have a very high number of targets that we need to detect in order to answer our experimental question, and this might exceed the limits of our flow cytometer. So for example, if our cytometer can detect only 10 colors and we need to detect 20 cytokines, this is not going to be possible. Also, we might have unused sample fractions. For example, we might have a plasma or serum that we haven't used for our experiment that might contain interesting biological information. It might also happen that the flow markers that are available are not covering all the information we need from a particular cell type. So we need to delve a little bit more into the function of the cell, how these cells are producing cytokines to answer our biological question. And also we have to consider that sometimes the antibody availability for some targets is reduced in the market, so we need to take different avenues to detect our desired cytokines. In all these cases, the flow experiment can be combined with other techniques, so these little friends that can help us achieve our goal and address our question. And as I mentioned before, the immunoassays will be coming to the rescue in this type of situations. And at Thermo Fisher Scientific, you can find multiple options, such as build-it-yourself immunoassays, where you can detect the antibodies, protein, substrates that you want to use to address your question in a very tailored and specific way. You can also check the different options for uncoated and coated ELISAs. For quantum assays, where we have a low sample consumption and high sensitivity protein detection on qPCR systems, and also some assays that might be of a very special interest in the situations we presented before, which are the Procartaplex and the quantigene multiplexing assays. In this type of assays, we can measure multiple proteins or RNAs simultaneously in one well on Luminex systems. So this can come very handy when we need to detect a very large number of targets. In this regard, we are bringing a couple of examples of how these multiplexing assays can be very helpful. In this case, you can see how we isolated PBMCs. And from here, we took the PBMC fraction and discarded the rest of the fraction and simulated the PBMCs with multiple molecules such as PHA, CON-A, or LPS. After simulation of the PBMCs, we can separate this into the cell lysate and the supernatant. We can take the supernatant to quantify proteins using our Procartaplex assays, and we can, for example, perform an 80-plex panel addressing 80 different targets. Or from the cell lysate, we can perform the gene expression analysis with the quantigene assay, again, reaching 80 different targets. These assays are also highly flexible, so we can address in a lot of detail the immune response at differentiating a different type of targets according to the experiment question we want to address. This type of multiplex immunoassays are also very important in the CAR T-cell research workflow, coming back to our CAR T-cell topic. As you can see in this diagram, there is a very important part in the workflow when we are generating CAR T-cells, which is to address the function of these cells and analyze how they behave when we co-culture them with tumor cells. If they kill effectively the tumor cell, and also the cytokine profile they are secreting to analyze their behavior in more detail. And this is something we can achieve with this type of multiplex immunoassays. We can identify, as I mentioned before, up to 80 different targets. So this allows us to understand a little bit better the cytotoxic profile of the CAR T cell, and also, on the other hand, to check if the cytokine production is excessive or even triggering detrimental effects for the patient. So this is something that we will need to target, as we will need to avoid any side effects and excessive immune activation before any CAR T cell can be used in a patient. With this, we have finished our cytokine highlight for today. So I would like to thank you all for your attention. And before we jump into the next section, I will rather highlight very quickly our flow cytometry panel design service. So just as a reminder, this is a free service, so no purchase is necessary. And we are here for you to support you in any flow cytometry panel needs for any type of instrument, no matter big or small. If you are interested, we are always happy to help, and please feel free to contact us by scanning the QR code or at thermofisher.com/LTF panel. I will also post our contact details in the Q&A section. Thank you so much, and I will hand over to Mehak for a brief communication.
Yes. So thank you so much, Andrea, for this informative session, and thank you all again who have joined us today. And before we move to the Q&A session, we'll also like to leave you with some quick resources that might be helpful for your learning and exploring, your flow cytometry journey. So here you see a snapshot for the Thermo Fisher's flow portfolio designed to support your entire workflow from sample preparation to analysis. When you are just getting started or working with high parameter panels, we offer a wide range of trusted reagents, antibodies, instrumentations, everything from sample preparation to the instrumentation. And if you'd like to explore further, feel free to visit our website, thermofisher.com/flow. Our product info, application guidelines, and all the helpful resources are there. And it's a great place to dig deeper at your own pace. And the next, I would also like to introduce, and don't forget to take advantage of any special offers or helpful resources when we have available. You can just scan these QR codes and check out our current promotions or get a quote or connect with one of the technical support teams. Yes. Okay, now we can move to the Q&A session. And just a quick reminder about this: to submit a question, click the Q&A icon at the top of your screens. Uh, and to ask a question live, you can use the raise hand feature and we will unmute you. And in the meantime, you think about your question, maybe I can get started. Something, Andrea, that I have wondered myself, if you can't find a good antibody for your cytokine or protein, can you walk us through some of those other ways you can still measure its production? So can you maybe briefly comment on it in the meantime?
Yes, absolutely. Thank you so
Yeah,
much for the question. And
yeah,
actually, this was one of the focus areas that we wanted to highlight today. As we mentioned before, we might get circumstances where antibodies might not be the best approach. Maybe there is no good antibody in the market, or this is something that doesn't have enough sensitivity to detect our target. And then we can use antibody independent methods, let's say. For example, one method we can use via flow cytometry that was not mentioned in the presentation is prime flow. So with prime flow, we can measure directly mRNA levels in our cells via flow cytometry. So instead of measuring the protein itself, we can measure the mRNA expression, which can, for example, give us enough sensitivity to detect this desired target. And going back to the presentation itself, sorry for the back and forth. Here we have also the quantigene assay option, which is also antibody independent, and this is a gene expression analysis that might also help us detect our desired target independently of the protein expression.
yes. Okay, thank you, Andrea, for the comments. Maybe we wait for others to think.
Let me go back to the thank you slide.
Yeah, and while we wait to see if any more questions come in, maybe in the meantime, I can ask you another one from my side as well. So I know a lot of us are excited about, spectral flow cytometry, what it can do with so many detectors available now. But I'm curious, there are still real limitations when you're, doing cytokine-driven experiments. If so, where do you usually run into the trouble? If you can comment on it.
Yes, thank you. So as you might have guessed already from my role, I really love flow cytometry, and this is very dear to me, but we have to realize that this has limitations. And although spectral flow is really advancing a lot and allowing us to measure more and more targets every time, going back a little bit into the discussion we mentioned before, sometimes we just have a sensitivity threshold that flow cytometry cannot surpass. So for example, sometimes by a flow cytometry, the detection limit we have for particular cytokines or proteins is not sufficient to detect them. So we need to use another method. So even though we can measure 50 parameters at the same time, the sensitivity might just not be enough for certain cytokines or proteins. So we need to go to an immunoassay or something else that allows us to have a more sensitive detection limit. And also in spectral flow, something that we always like to highlight is the effects of spreading error, fluorophore interaction. And although we are getting better every day having new fluorophores, we need to be always very mindful of co-expression. So sometimes spectral flow might not be the best one to do a very in-depth cytokine profiling in the same cell type.
Okay, thank you for your comments. Anyone, any other questions? Maybe I want to ask another thing.
Sure.
How reliable are the cytokines response we see in vitro compared to what actually happens in vivo or in ex vivo samples? I imagine there can be some differences, but I'd love to hear, your thoughts on it.
Thank you. This is actually something quite interesting, and we could have a very long discussion about it because in the end it will be something very particular to a given experiment. So although when developing products we put a focus on trying to mimic as much as possible the in vivo responses, having the full activation profile, the full phenotype profile that a cell has in vivo in vitro is sometimes just not possible. It depends also on the system you want to analyze. So for example, if you want to analyze something very straightforward, like the activation by a couple of proteins, this might be easier to replicate in vitro compared to a very complex system like a multifactorial disease where multiple activation and inhibition pathways might be ongoing. So this is something that might be necessary to address case by case. But I would say the simpler scenarios would be easier to mimic in vitro rather than more complex.
Okay, thank you. Anyone, any other questions? Last chance. Okay, that I think looks like all the questions we have like for now. But thank you again for joining us today, and a big thank you to Andrea for sharing all the valuable insights and the nice presentation. We hope to see you on our next month's session, where we'll be covering the topic crafting a gating tree, unveiling co-expression patterns using an imaging enhanced flow cytometry. And yeah, I wish you a great day ahead from my side. And if you think of any questions, just feel free to reach out to us at any time. And I wish you a great day and a nice, long, extended weekend. Then thank you again for your time today. Yeah. Bye bye.